He's an assistant professor and separate scholar in cancer research in the Department of Bioengineering at Rice University. He's also a co-founder of Sigilon Therapeutics, a Cambridge-based pharmaceutical company that discovers and develops immune-privileged implants for treatment of chronic diseases. Dr. Weiss received dual PhD in material science and engineering and nanotechnology from University of Washington. Next, he completed his postdoctoral work with Professor Bob Langer and Daniel Anderson at MIT and Harvard Medical School. Over his career, he published more than 50 papers in top-notch journals, including Nature, Nature Biotech, Nature Materials, Nature Medicine. Then he's an inventor of 20 pending or awarded patents. He commercialized 3 separate biotech companies. He's won numerous awards, including a $2 million separate scholar in cancer research award from the state of Texas, and he was recently named one of the MedTech Boston's 40 under 40 healthcare innovators of 2017. Floor is yours.
Thank you so much, Mehmet. Appreciate the invitation and the opportunity to present. Looks like we have a great turnout today. So thank you all for coming. You know, the opportunity for us to tell you a little bit about some of the work that we're doing in developing cell-based therapeutics for a number of different diseases. I guess before we get started, I just wanted to provide you with a list of my disclosures, and any perceived or real conflicts of interest are managed by our dean's office here at Rice University in the School of Engineering. So one of the things that my lab is interested in, and our vision, is to really think about the next generation of drug delivery technologies that need to be invented. And you know, when we think about the pharmaceutical industry, there's been great progress with small molecule drugs. And in the last 3 decades, biologics have become such an integral part of management of many diseases. And in particular, when it comes to chronic diseases, then there's a need to provide these biologics routinely, but they always have to be injected. So what's been emerging is this concept of gene therapy, which has had some successes, but has some limitations in terms of scaling doses, the ability to redose, the ability to treat juveniles. And what we think is a unique opportunity is to innovate cell therapies, and in the form of encapsulated cell therapies, where allogeneic cells could be produced, and these could be drug factories that are then implanted into patients to produce drugs on an as-needed basis. So we're broadly interested in this concept of cell-based therapeutics, which is the utilization of this approach for drug delivery for chronic indications, for indications where spatial temporal control is going to be critical, and where cells can sense and respond. So the benefits of this is that, you know, by using encapsulation, we can create these isolated systems whereby our cell factories could thrive and, you know, gain nutrients from the host and produce different types of biologics that then could be secreted and produced into the patients. We can program the durability, deliver drugs either locally or for the serum, and this has, you know, different patient benefits depending on the indication that we go after. So the first part of my talk is some of our efforts geared towards innovating this solution for the treatment of type 1 diabetes. And for those of you in the audience that may not be as familiar, there's 2 types of diabetes. There's type 2 diabetes, which is primarily a lifestyle disorder. And then there's type 1, where by reasons we don't quite yet understand, the body's own immune system mounts an immune attack against the insulin-producing cells of the pancreas. These are termed beta cells. And as these beta cells are killed off by the patient's own immune system, then you know, the patient loses the ability to regulate their blood glucose. Instead, they have to persistently monitor their blood glucose through handheld glucose meters or continuous glucose meters, and then administer insulin exogenously to correct and maintain a physiological level. And this is quite burdensome to do properly. And most patients actually are not able to do this correctly, and only are able to be at the physiological right levels for several hours throughout the day. And this can have consequences long-term health of the patient. What's available today is this concept of pancreatic islet transplantation, whereby you can take a recipient's pancreas, digest it down, and reintroduce it into a type 1 diabetic patient. This requires immunosuppression, and it takes about 2 to 3 donors to actually treat one recipient. And so this procedure is not widely used because of the limitations in the availability of organs and the need for immunosuppression. And each year there's maybe about 2,000 patients that benefit from this approach, whereby we have millions of type 1 diabetic patients. But what's exciting is that because many patients each year get this procedure, what we now understand is that, it does work. And when the patients receive these transplanted cells, they are able to restore their glycemic levels. And there's patients now that are maybe 15 years out and still insulin independent, meaning that the cells that they received as a transplant have kicked in, they're sensing the blood glucose, and they're secreting insulin on an as-needed basis. So the concept works, but what we really need is ways to get rid of the necessity for immunosuppression. And also, we need to leverage cell forms that are replenishable so that we don't have to rely on transplanted organs. What's been exciting is that in the last, I would say, 10 to 15 years, the scientific community has made great progress in figuring out the recipe that's needed to take a stem cell in vitro and in culture, basically guide the differentiation of these stem cells towards beta cells. And with a stem cell supply of beta cells, then the need for organ transplantation becomes unnecessary. And we can actually produce these at large scales and generate as much beta cells as we need. And some of the pioneering work of Doug Melton and others in this area have shown that this could be made on a commercial scale. And this provides an opportunity for us now to leverage these different cells with encapsulation technologies to achieve long-term correction. So when you have the cells, what we need to provide is a way to shield them from the host immune system. A type 1 diabetic patient, their immune system is trained to attack any insulin-producing cells. So this concept is taking novel polymers, providing a matrix around the beta cells that excludes the immune cells from actually being able to contact in order to kill these insulin-producing cells. If this matrix is porous so that oxygen and nutrients can get in and out, and the therapeutics, which in this case is insulin, can get out, this could be transformative for the patients. It alleviates the need for having to do immunosuppression. And one of the materials that people have been exploring for this application for many years is alginate polymers. The reason why alginate is preferred is because you can create a hydrogel matrix around cells using really gentle reactions, such as chelation. It naturally chelates with divalent cations, such as calcium or barium, and it forms a stable structure around the cells, which is actually a porous matrix, such that the porosity is tuned to exclude immune cells and at the same time allows for diffusion of nutrients and oxygen in and out. But most of the ways these encapsulation strategies allow you to, create these bubbles around your islets, and you can do this while maintaining more than 99% viability of the cells, which is a key requirement of this. Now, of course, alginate has been around for many years, and this concept of encapsulated alginates, using alginates for cell encapsulation, has been around for a while. In fact, the first publication on this concept was in 1980 by Lim and Son out of Edmonton University. And one of the things that they demonstrated at the time was you could take cells, put them in alginate hydrogels, and they demonstrated that, what normally would happen is if you have unencapsulated islets and you implant them into these diabetic mice, then they get rejected quite rapidly. Within a few days, the mice's glycemic correction improvement goes away. But through encapsulation, you can actually protect against that T-cell mediated attack by the immune system. But one of the challenges that they also identified is that, a couple of weeks in, because alginate is not a material that has any abilities to withstand foreign body recognition, the host would recognize the alginate polymer as a foreign body and mount a fibrotic response against the material. And as this fibrotic overgrowth built up on the alginate, then the cells on the inside, died off. And so this concept, while it works, doesn't last very long to be a therapeutically viable strategy. And over the years, there's been a lot of work trying to improve this. And there's been a number of trials and different approaches that have gone all the way to human patients and non-human primates. And the outcome is always the same, that, this fibrosis challenge is what really prohibits this from working. So one of the goals for our program was figuring out a way to reduce the fibrotic response to the polymer, creating more biocompatible hydrogels to achieve, improved clinical outcomes. And what we know is that, alginate, its composition is such that the host immune cells, in particular macrophages, recognize alginate polymers as a foreign substance and basically attach to the surface of these capsules and become activated and release cytokines, which further recruits myofibroblasts and other immune cells. And ultimately, after a few weeks, you end up with this matrix of extracellular matrix that has deposited onto these hydrogel capsules that now is a barrier to diffusion of oxygen and nutrients into the capsules. And so your transplanted cells essentially starve and are killed off. And that's when the therapy stops working. So we thought about different ways to improve outcomes. And one of the areas we particularly focused on was creating an anti-fibrotic alginate. And of course, there's a lot of different approaches that have been postulated in the literature for addressing this. But one of the things that we quickly realized is that it's hard to know which one would work and what the best strategy is. And there really isn't, you can't go to the literature and say, okay, if we do this modification, then this will for sure work. So we decided to take a much more broad approach and kind of do something similar to what the pharmaceutical industry typically does, in that when they have a phenotypic outcome, if they're looking for a specific outcome, they would actually explore a lot of different libraries of different compounds to see which would work better. And we actually thought maybe we can apply the same exact approach to the discovery of novel biomaterials, which could alleviate foreign body response recognition and have better outcomes in terms of fibrosis. So we generated a library of alginates, which were chemically modified. And this was a very broad screen. And at the time, we didn't have any biases about which molecules could work. We tried to go as broad as possible. We had access to automation. And this was some of the work that I started in my postdoc that allowed us to build large libraries of these different alginates. And the hard part was, once you have a lot of different combinations of materials, how do you determine which is a good versus bad material? And one thing that we explored, one possibility is to implant material in a high throughput model where we can actually put many different materials in the same animal and look for signs of inflammation. This is similar to an allergy test, where when you're trying to figure out what things the body might react to, you might administer a little bit of it underneath the skin of a human and see where you see reactions. We applied the same exact principle to testing all of our formulations, and we could, in parallel, actually test many materials on the back of a mouse that are spatially separated. And we had this ProSense method, which allowed us to quantify the signal, and then the lead materials would then be tested in different implant sites as individuals and also with cells to evaluate whether we could actually achieve better clinical outcomes, better therapeutic outcomes. So this is a bit on the design of the library. These are combinatorial reactions whereby you actually build the molecule off the alginate backbone. And this allows us to create a very diverse set of chemical modifications. And the goal was really to produce as much diversity in terms of amines, amides, trizoles, alcohols, as much chemical diversity as possible, and perhaps identify trends as far as these type of modifications elicit improved alginate activity, and then, based on that, design second and third generation libraries and ultimately find materials that would perform really well. I mentioned that we had this sub-Q assay where we would implant, materials in discrete positions, and these are, 300 micron spheres. So once you inject them into these positions, they don't really go anywhere. They stay exactly where you injected them. And we wanted to evaluate how the immune system of the mouse was reacting to these materials. So we developed this ProSense imaging approach, whereby you can inject a dye, which is normally chelated, but in the presence of caceptins, and caceptins are secreted enzymes by macrophages and neutrophils. So wherever you have activated macrophages and neutrophils, you would get a fluorescent signal that would evolve. And that would allow us to quantify exactly how much response the immune system is orchestrating against a given material. And this is sort of what it looks like in practice, in that you have these materials in different spots where there's a lot of inflammation, which correlates with fibrosis outcomes, poor fibrosis outcomes. You see a lot of signal, but if the material is good, you actually don't see much signal at all. And this correlates with the kinetics and composition of the immune response in terms of macrophage and neutrophil activity. And so where we don't see signal, we do correlate that with long-term improved outcomes in terms of fibrosis. And so this is actually the results from a subset of the screens that we did. And each of the materials are identified by a number. So this is formulation 1 all the way out to, in this case, we did 774 different formulations. And one of the things that you notice is, the base material, which is the unmodified alginate, produces a particular fluorescence response that we set at 1. And we noticed that many of our modifications actually exacerbated the immune response much more significantly. But we were excited about these formulations down here, which were materials that actually did not provoke much of an immune response at all. And so we wanted to learn more about what those are. And these are the formulations of the leads. You can see that they have a particular chemical composition, and what's common among all 3 of the leads is that they have this triazole group, and there's the PEG linker, this triazole group, and then there's what we call the cap. And what's interesting is that these 3 performed much better than any of the other modifications we tested, and also allow us to reduce foreign body response and fibrosis of the alginates by more than 90%. And these were the ones that we wanted to move forward with further. And we could take those same small molecules and actually apply them to a number of different types of surfaces. For example, here's a study we did with polystyrene microspheres. And if you're familiar, polystyrene is the material that we use for tissue culture. It's a highly inflammatory material. It's a highly fibrotic material. And when you implant it without any modifications, within 2 weeks, the mouse's immune system orchestrates a robust immune response, and all of the beads get engulfed in this scar tissue. When we take that small molecule and we apply it to the surface of these polystyrene beads and we implant them, the material looks like it did before it went in. So dramatic reduction in immune recognition, fibrosis, and we can apply this to a number of different substrates. So we wanted to understand, why we're getting these impressive results. So we did this study, and this is in mice where all the macrophages express green fluorescent protein. This is in the intraperitoneal space. Our implants are in magenta, and in this case, they're unmodified. And we evaluated how the immune cell perceived the implants using this technique. And this is about 6 hours of imaging that's been condensed to like a 30-second loop. And one of the things that we notice is, there's quite a lot of immune cells there when there's an unmodified material. The immune cells have adhered to the implant, and they have recruited additional immune cells to come in. You can see the immune cells are quite active. There's a lot of them there. They're more and more coming in. And really, this is activated immune response mounted against the biomaterial. And so this is the scar tissue formation that we're trying to address. What's fascinating is that when we have the same exact situation, but the material has our small molecule coating, you can see that there's immune cells there. They're present in healthy numbers, but the immune cells actually are crawling on the surface of the material, but they're not mounting, they're not adhering. And when they don't adhere, they don't become activated. And they're just harmonizing with the immune system. And there is not a foreign body response that gets initiated. And these small molecules we can apply to different surfaces and really probe this mechanism further and understand what it is about these molecules that are giving this preferred response. And so we switched to working with discs of different medical devices. So silicone, PDMS, these are polymers that are widely used by the medical device industry. And we wanted to see, what would happen if we attach our modifications, our chemical modifications to the surface of this. And we first quantified and showed that we have the coatings on there. And we implanted these into mice. And again, when you have the chemical modification, there's no immune response and no scar tissue formation. When it's just the material by itself, you get a pretty nasty scar tissue formation. And this is true for a wide range of polymers. This is polyethylene that's been treated with a small molecule. Each of this went into a different animal. And you can see without the coating, you get a lot of scar tissue formation. With the coating, they come out crystal clear, no immune response. And we know the fibrosis outcomes from the studies that we did, but actually probing this further and using a mouse model where all the macrophages and myofibroblasts express green and red fluorescent protein respectively, we're able to see what it actually looks like in real time. The material is in white, and this is the immune response that's mounted to the unmodified material. So you can see a lot of macrophages and myofibroblasts on the surface of the material, but also adjacent to the implant, many millimeters away, we see a lot of inflammation, a lot of macrophages that are present there. And this would be a cause of significant pain and discomfort to a patient that would have this type of an implant. By contrast, when you have the modified PDMS, you actually see very few immune cells, no signs of inflammation, and the material is better tolerated by the host. And these type of modifications have drug-like activity in that we've done experiments where we titrated the amount of the molecule onto a surface. And we noticed that at a certain point, once without the modification, you get this immune response. As you apply the small molecule, it starts to work, but it works less and less until you get to a particular density of coating where you get this robust immune response, and you can quantify it, and you see like a dose response function, which correlates with drug.
Omid, you froze. I can't hear you.
molecules. Yeah.
Okay.
Okay, there we go. Okay,
It's
great.
better now. We can hear you now.
Okay, great.
We lost you
Awesome.
in one slide, but you want to go back one slide?
Go one slide. Okay, yeah. Did you guys
Yeah, okay.
hear this?
Yes.
Okay, yeah. So basically, the conclusion of this slide was that our small molecules have drug-like activity in that you see a dose-response correlation between the amount of small molecule presence and the foreign body response that are the outcomes. And our results are actually durable in that the small molecule, when it's applied, it has long-lasting effects. And in this study, we went out for 90 days, and you can see that each of these discs went into a different animal. And with the modification, you're actually able to address this foreign body response. And the other reason why we think they have drug-like activity is because, again, these discs, when they're unmodified, they initiate an immune response. When we attach the small molecule to it, they no longer the body doesn't recognize them as foreign, and these discs come out clear. But if you look at this oxygen group, when we created an analog whereby we moved that oxygen group to this position, actually it entirely killed the activity of the molecule. And what's fascinating about this is that these two molecules actually have the exact same physical properties because they have this identical number of oxygen, nitrogens, and carbons, but it's just the specific molecule activity that's giving rise to this impressive response that we see, similar to how drugs work. So an analog of it failed essentially. And all we had to do to break it is just move one oxygen group, and the whole thing lost all activity. So it's pretty exciting that we stumbled upon the ones that worked, kind of fortuitous in the sense, because it seems to be very specific ones that give you this impressive results. And of course, this has implications to the design of a wide range of different medical devices. There's a lot of devices that go into the body that today fail quite frequently. One example is ventricular peritoneal shunts. These are systems where there's a part of it that gets implanted into the ventricles of the brain, and then there's tubing that's tunneled all the way down to the peritoneal space and allows for drainage of fluid from the brain to maintain a proper pressure in the brain. And unfortunately, in the pediatric population, where this is used in a subpopulation of patients that are born with congenital issues such that they can't regulate the pressure in their brain, these devices fail quite frequently. So 40% of them fail within 2 years, 98% of them fail within 10 years. And there's case studies, and our collaborators who helped us with this study, they indicated to us, they've got like 13-year-old patients that have had like 80 neurosurgeries having these devices constantly replaced. And the reason why they fail is because of the foreign body response that's mounted. And so we did a study with them, and with Medtronic, who normally sells these shunts, we actually acquired the exact same shunts that they have on the market today. And we took them and cut them up into smaller pieces so they can fit into rodents. And, we also observed even in the rodent models that, these devices become occluded because there's that foreign body response that is orchestrated against it. But when we apply our small molecule coating, they don't become recognized as foreign, they don't become occluded. And this is durable. So this is, again, a 90-day study where we show that, they become free-flowing still. And of course, there's a component of this device that goes into the brain, which is a different implant site than the peritoneal, where we've been testing these so far. And even in the brain, where the immune cells that are responding are microglial and astrocytes, we see a dramatic reduction of the inflammation when there's the coating applied to it. So regardless of implant site, this actually works very well. We're now working with Medtronic to actually test this in an efficacy model where you can actually create hydrocephalus and show that this does lead to better outcomes. we've also tested our hydrogels in non-human primates just to show that, this does translate to larger animals. And this is after 6-month implantation. And at the beginning of my talk, I provided some example images of various studies with alginate capsules that have been done in non-human primates, and they all got fibrosed. And this was really one of the first evidence that you can now create hydrogels, alginate hydrogels, implant them in non-human primates, and be able to retrieve them fibrosis-free. So this was very exciting for us. So now that we had the right material optimized, going back to the cells, we wanted to load them up with the appropriate cells to come up with a solution for type 1 diabetes patients. And there's a lot of cell sources available, but we're really excited about the stem cell derived sources that are now available for use. And so we work with Doug Moulton's group out of Harvard, who had this protocol for taking embryonic stem cells and differentiating them. And when we used those cells in our capsules, we first tested them in rodents in different densities and with and without the coated hydrogels. And when we implanted them, without the coating, the material that was essentially what's considered to be clinical grade, available, and had been to the clinical trials, it only worked for a couple of weeks, very similar to what Lim and Sun saw in their studies. When we have the modification, we're actually able to see a much durable response because the material is no longer getting fibrosed, and so the cells are able to last much longer. In this case, we went out for 90 days. And this is what the capsules look like when we retrieve them. With the modification, the myofibroblasts, which stain red, don't appear on these capsules. And when you have different sizes of capsules, small or large, you can see that these myofibroblasts appear and the islet cells just start to die off. And then we profiled the immune response to these capsules, and also these had the cells in there. And this was a xeno model where you have human cells inside of these immunomodulatory coatings. And what we noticed is that, the modification suppresses the macrophages and the neutrophils, and those are involved in the foreign body response. So that was expected. But the other thing that we noticed is that they actually suppresses the activity of the cytotoxic T cells and the B cells. And this is important because these cells could secrete molecules that could be detrimental to our encapsulated cells. But because we're reducing the overall inflammation and fibrosis, those cells are also not activated as much either. And so we wanted to do, I guess the killer experiment, which is, can we cure a mouse, a diabetic mouse, for the duration of its life, essentially? And a diabetic mouse usually lives around 6 months. And so we did this study, and we demonstrated that, yeah, we were able to maintain glycemic correction, not as good as a healthy mouse, because there's still some things that are not exactly there, in terms of connection to the nervous system and whatnot. But, it's going to be much better than patients kind of checking their own blood glucose, administering insulin on an as-needed basis. And that's sort of the competition that exists today. And after we took out the capsules from these 6-month studies, we could still see viable islets in there, and they hadn't de-differentiated. So it was exciting to see that, the concept works in rodents. But other people had shown great rodent data in the past as well. And we were excited about really translating this technology and thinking about, getting, how do we get this into humans? We thought that we had, moved the needle significantly. And so when we started talking to potential investors, they basically were like, well, a lot of people have great mouse data, but then they always fail in humans. And, in fact, there had been many trials with alginate capsules over the years, which have been done in human patients. And these were things that looked great in mice. They went to human patients and they failed, and they saw these dramatic foreign body responses to the alginate. We actually contacted the investigators of those trials, and we said, hey, you guys had great materials that look promising and failed in humans. We think we have great materials that look promising. We haven't gotten to humans yet. Let's do a side-by-side comparison of all these different formulations in non-human primates as a bridging study to determine whether we'd move the needle significantly enough to warrant additional human clinical trials. And when we did this comparison, we noticed that, in the non-human primate model, our capsules are still coming out clean and fibrosis free, but all of those other ones that look promising in mice actually don't work in non-human primates either. So this helped kind of provide a link between, where the field was in terms of stuff that wasn't working in humans. Now we show that, yeah, those don't also work well in non-human primates, but with the modification, we're able to see great results in non-human primates with our formulation. And this actually in non-human primates results in encapsulated cells remaining viable. And we also identified perhaps a preferred implant site, which is the bursa mentalis. It's a sac that actually exists in the IP space. It's covering the liver, and it's accessible in that you can actually put capsules inside of this sac. And we found that we even get better outcomes when we take our modified alginates with the cells and we put them into this area. And then, this is 4 months later without immunosuppression, we take out this, and these capsules actually all look translucent, no fibrosis. And when we assess the viability of the cells on the inside, we see that more than 80% of the cells are viable and are able to remain functional with no immunosuppression in non-human primates. And the cells are able to respond to glucose challenges and secrete insulin, which is really exciting and provides an opportunity now for future clinical investigation. So we started this company called Therapeutics, and we formed a partnership with Eli Lilly. And so now there's even a bigger team that's really pushing this forward. And we're on track to move our diabetes program into the clinic. But we have another program that actually is already in the clinic. And, this fall, the company IPO'd, and we actually dosed our first set of patients for another indication, which is hemophilia. And in this case, the cells are producing factor VIII, which is necessary for treating hemophilia. And what's exciting is, these patients, these capsules, which were administered to this human patient, we now, it's sort of validation of all the preclinical work that we did in terms of, optimizing the types of cells that go in here and the chemistry and the sizes of these capsules. And so to us, this is, pretty exciting to see all of that come together in terms of this clinical trial that got started. And of course, for the patients, this means freedom. This means not having to inject yourself regularly. This is true for a lot of these chronic disorders, and we've been focused on diabetes, but there's so many other chronic disorders where this can have benefit. And, for hemophilia, when you consider what a child who has hemophilia has to deal with, this is 1 month supply of the factors that, they have to inject into themselves on a daily basis. And what we can do is really translate that to a small implant that gets administered to the IP space. And now the drugs are produced in the patient on an as-needed basis, as opposed to produced in the bioreactor, put into vials, and then shipped to patients to administer. So we're hoping that this is going to really transform how biologics are used for chronic diseases. And we're excited to see how these trials end up. So far, what is publicly available is that, the first 3 patients are doing well and that the levels are detectable. So we're really excited about this. And we're also investigating other types of lead formulations in terms of, now that we know that we have these trizoles that work well, we've come up with ways to even expand our screening capabilities so that we can actually test many more formulations simultaneously. We've used this barcoding strategy where we can get a lot more information about the material that we're implanting. More material can be tested simultaneously, but also we get additional information about the particular types of immune responses that are being mounted against these beads. And so we have a project that's focused on this. And through this, we've even found better leads and the ability to test, many materials simultaneously in non-human primates at once. And so that's, we've even found new molecules that could actually work equally well or perhaps better than some of our original leads by really digging further and pushing this approach even more. And I'm going to switch gear and just quickly talk about another application of these encapsulated cells, which is for cancer immunotherapy. And at the beginning of the talk, we talked about how the goal was coming up with materials that don't provoke an immune response. But in the process of doing that, we actually discovered a lot of materials that are very inflammatory. And so one of the things when I started my lab was to think about, can we leverage them to provoke tailored immune responses in ways that we want, to basically aggravate the immune system to start fighting cancer cells. And that's really the basis of this project that I'm going to describe to you now. And when we think about immunotherapy, it's a combination of having the right signals there for the immune cells, and cytokines are one way where you can regulate how the immune cells are communicating together. And we started to think about novel cytokines that we could have cells that are engineered to produce these cytokines, but also pair them with super inflammatory materials to really aggravate the immune response in terms of being able to produce an effector immune pro-inflammatory response that is necessary to basically take a cold tumor and turn it into a hot one. And so for this purpose, we actually focused on the materials that are really inflammatory, the other end of the scale. So these are good for chronic use. These are good for acute use where you want to initiate an immune response. And so we created hydrogel capsules and showed that we could have different cytokines produced from these materials that are really inflammatory, and create local concentration gradients of these cytokines through feedback loops, whereby, within the tumor microenvironment, and this is using some innovative synthetic biology that I won't go into, we can create local concentration gradients of these cytokines so that they maintain a local high concentration, but then systemically, there's actually no cytokines, because these cytokines systemically can be toxic, but locally they can be beneficial in terms of activating the immune response within the intraperitoneal space. And the goal is really to create local high concentrations to provoke effector T cells to become activated. And once those are activated, they will find the cancer cells and kill them off. But you have to get the concentration right. And that's part of the innovation that we trying to incorporate by using engineered cells that have feedback loops that maintain particular concentrations locally. And we're able to demonstrate, and again, when we use the inflammatory materials, the material itself causes an immune response, which is what we want. And so we're able to maintain these concentration gradients for a determined amount of time within that cavity, and locally deliver the drug where it needs to be in the tumor microenvironment. And so we did this study with ID8 tumor mice, where this is a model of ovarian cancer, and we showed that, based on the dosing, we can actually achieve a dose response effect in terms of treating the mice of ovarian cancer. And this is a particularly tough model that has really seen not a lot of success with immunotherapy, but our results suggest that, when you combine the right formulation of the cells plus the right cytokine and the material, we're actually able to eradicate these tumors in these mice that don't respond particularly well to immunotherapy. And this mechanism mechanistically is guided by the presence of T cells, because when we get rid of the T cells, then the therapy stops working. And we know that those are the preferred populations that we're expanding. And we can actually try this in other tumor models. This is another model that we've used for colorectal cancer, and we see the same exciting results in that, our therapy can help eradicate these tumors that are particularly difficult to treat. And what's exciting is that once we treat the tumor, because the way that this concept works with immunotherapy is you're really training the immune system to recognize the cancer. And once you have treated it locally, then you protect it systemically as well, because now the immune system knows what the cancer cells look like, and they go throughout the body and find them. In particular, we did a study where we took a local where we treated the peritoneal disease, and we showed that actually that affected the systemic disease as well. In that when we tried to grow the same tumor in another part of the animal, it was rejected because of the memory T cells that had formed in response to our therapy. So yeah, with that, I know I'm getting short on time. I just wanted to thank a lot of the work. This sort of transcended from started my postdoc, and then I went to Sigilon Therapeutics, and I was there full time for a couple of years. And now I have my own lab, and so I wanted to thank some of the my fellow co-founders of Sigilon Therapeutics, Bob Langer, Dan Anderson, Arturo Vegas, Jose Oberhauser. Together we started this company, and it's exciting to see the progress for chronic diseases. We're all looking forward to seeing the human data that should be coming out over the next year or two. And of course, the members of my lab and our funding sources. And with that, I'm happy to take questions.
Thank you for the great talk, Amit. Several questions came in. One of them is, great presentation. Thanks for sharing the question. The released insulin contains C-peptide. How do you cut that to make the pre-insulin active?
Well, so that part, the biology does for us, because we're taking the beta cells, they're stem cell derived islets. And so they're basically acting like beta cells would, in that they have the right machinery, they process the insulin the right way, they sense glucose and secrete them.
All right.
If I understood the question correctly.
Yes. Although immune cells, T cells particularly, cannot come in contact with encapsulated cells, the latter may secrete molecules that can be immunogenic, such as soluble allogeneic HLA molecules. Therefore, for encapsulated cells, therapeutics, islet transplantation, for example, was the humoral response evaluated?
So one of the things that we noticed was that, if you get rid of the ability of the cells to directly contact, we are able to, get these cells to live a long time. I think that's a good point. And, I guess we'll see in the clinical patients what happens. But I guess in the studies that we've done so far, because we're testing human cells in animal cells, you would expect that those responses would exist anyway, because it's a mismatch. But our data shows that they do last quite a substantial amount of time so far. But longer term, we'll have to see in human patients.
All right. Can the biomaterial used for cell encapsulation modulate HLA class 1 or class 2 expression?
Yeah, that's a really interesting question. I mean, we haven't looked at that. I guess there could be, innovative ways to think about it. Yeah, could be a different axis to consider in terms of the function of the biomaterials.
How large can you make the alginate capsules, on the order of microns? Is there a maximum and minimum diameter?
I mean, I think, in the literature, people have made formulations that are as small as, 10 microns for single cell encapsulation. The formulation that we work with is actually a larger one. They're 1.5 millimeters in size. And we had a paper that we published in 2015 in Nature Materials that actually looked at the size and identified the 1.5 millimeter size to be preferred in terms of immune response and cell viability. But yeah, there's a range that could be explored for different applications.
Okay. How do you prevent the alginate degradation? Do you cross-link them constantly after the transplantation?
So the alginates, the way that, if we use the formulations we're using, they don't really degrade in the body. So we cross-link them with barium. They're pretty robust, stable. And we have studies that we've gone out for up to 1 year in non-human primates, and the capsules come out intact and they haven't, degraded at all. So I think, it's just using the right cross-linker, divalent cation. I think it is a problem if you just use calcium, that the formulation would degrade, but with barium it doesn't.
I see. Did you try to see why those chemistry are getting blind to immune cells from a biological point of view?
Yeah, I think that's a great question. I mean, we're probing the mechanism a lot more. we want to understand a bit more about how they work. That's sort of the next step. We've got a couple of students that are interested in it. We have some ideas, and as that kind of matures further, we hope to be able to share that in publications. But right now, we don't fully understand. We have some clues, but yeah, I would say TBD.
Have you applied this coating in blood, pro-thrombogenic or anti-thrombotic?
We haven't looked at whether they would have an impact on compatibility, hemocompatibility, but that would be an interesting thing to explore. Yeah, that's not something we've looked at so far.
Okay, somebody has an idea. Can you transplant memory cells from the one who has antibodies against COVID to someone else?
Yeah, that's a really cool idea, actually. It's something we've been thinking about, like, for example, if you can take B cells and encapsulate them and then administer them to someone that, is immunosuppressed, that can't produce antibodies on their own. But you could take a repertoire of B cells from a trained immune system and do a transplantation. And that's actually a cool idea that we've thought about. We haven't done it, but happy to collaborate with people if they're interested. But I think that would be a really interesting way to leverage this technology.
Did you evaluate whether inflammatory materials can induce autoimmunity if pro-inflammatory signal is too potent?
We haven't done that, and that would be interesting to look at. I mean, I guess depends on the patient population we're going into. Of course, for oncology patients, it might not be as bad of a problem. It's possible that they could lead to downstream autoimmunity, but that is a complication of immunotherapy anyway. Even with like checkpoint inhibitors or CAR T cells, that is one of the side effects, is people do tend to become much more susceptible to the autoimmune diseases in the future. But the immediate problem is, of course, to treat the cancer. But yeah, that's a good point.
What was the reason to modifying alginate to make it suitable for your purpose instead of trying other materials?
Well, alginate had favorable properties that made this practical, and we thought it was deficient in biocompatibility, so we thought we could fix that. Other materials have other challenges that could be problematic too. For example, cell viability upon encapsulation, if they have the right porosity, because you need to get the nutrients in and out, and some of those molecules are quite large in terms of molecular weight, so like some things like PEG polymers wouldn't really work. So yeah, I think it was just, alginate's great in many other ways except for this, so let's just try to solve this problem.
Great. How do you see these immunomodulatory biomaterials to be used in autoimmune diseases?
Yeah, that's a good question. I mean, I think there's a lot of opportunities there, in particular through modulating the host immune response. That's one approach. We're also considering, is there ways where we can train the immune system to become more tolerant? There's a lot of competing technologies to do that, including like mRNA delivery. But I think they're using the ability to localize the therapy and program the duration and expressions, provide us nice accesses to do immunoengineering in terms of engaging with the immune system in ways that could achieve those goals.
Okay. Have you thought about formulation of peptides into your encapsulation for intestinal disease?
Well, so we can certainly engineer cells to produce peptides of interest. I think, the formulations, because of their pore sizes, are not ideal for like biologic encapsulation by itself. But, I think there's opportunities to engineer cells to produce therapeutic peptides.
Did you ever think about using pancreas ECM gel for encapsulation of B cells? It is natural and safe, we think.
Yeah, that's a good point. We haven't, but that could be a really interesting thing to explore to see if the cells like it better. Yeah.
For the hemophilia application, what cells are used to deliver FEIL? Is the FEIL full length or truncated?
Yeah, so that's some of the work that Sigalon's doing, and I think there's some information about that in their publications. I think they are using the truncated one, and I'm not sure if they've disclosed the cells yet, but they're working with an epithelial cell that they've developed themselves.
I guess the final comment was decellularized pancreas ECM gel.
Yeah, so right, and that's relation to the ECM. Yeah, I mean, I think that's an interesting idea. Could be exciting to explore something like that. Yeah, we haven't done that yet, but it could be interesting to explore.
Awesome. Well, that's all the questions I have, Amit. Thank you for your time and the great seminar. I appreciate it.
Yeah, thanks for the opportunity.
Thank you, Amit. This was fantastic.
Oh, thanks, Ali. How are you?
Meet up soon. Yeah.
Yeah, definitely. Looking forward to in-person conferences again.
Wonderful.
Yeah, thank you. Appreciate it.
Have a good one.
Thanks, everyone.
Bye-bye.